Hello everyone, myself Sidharth Chandran from Central University of Tamil Nadu, currently pursuing my masters in microbiology. And today in this session, we are going to have a discussion on the topic factors affecting bioremediation. So bioremediation is a process where we use microorganisms such as bacteria and fungi in order to degrade or decompose harmful pollutants present in our nature. So here what we do is that we convert these harmful pollutants or contaminants into less harmful or completely harmless substances using microorganisms. So the benefits of bioremediation is that when compared to our traditional cleanup methods, it is more environmental friendly because we don't use any harmful chemicals or anything, and it is more versatile and cost effective. Now let's see some let's see few examples of bioremediation. So oil spills, contaminated groundwater, and soil pollutions are three examples of bioremediation. So now coming to the types of bioremediation. Basically, bioremediation techniques are of two types: in situ bioremediation and ex situ bioremediation. In situ bioremediation is a process where we conduct the process of bioremediation at the site of contamination itself. Whereas in ex situ bioremediation, we have to transfer the contaminated material into a controlled environment where it is broken down by microorganisms. So in situ bioremediation is itself of two types: intrinsic and engineered in situ bioremediation. So in intrinsic in situ bioremediation, we solely relies on the microorganisms that are already present at the contaminated site. Whereas in engineered in situ bioremediation, we add by microorganisms, or we add nutrients, or we modify the conditions at the contaminated sites in order to speed up the bioremediation process. So it's of many types like bioventing, biosparging, bioaugmentation, biostimulation, slurping, etc. So for example, bioaugmentation is a process where we add specific microorganism to a contaminated site in order to speed up the process of bioremediation. In the case of ex situ bioremediation, it is mainly carried out in two phases: slurry phase and solid phase. So in slurry phase, we use bioreactors, mainly most of the times stir tank bioreactors. In solid phase, we mainly use four methods: land farming, biocomposting, biopiling, and biofilling. Introduction part is done. Now moving on to the main part of the presentation, that is factors affecting bioremediation. So the bioremediation process is mainly influenced by factors such as environmental factors, biological factors, chemical factors, physical factors, technical and operational factors, and site-specific factors. Out of this, in this session, I will be discussing biological factors, technical and operational factors, and chemical factors. We will be seeing the biological factors in detail. So microremediation. Microremediation is a term used to define the process where we use microorganisms in order to treat these pollutants or contaminants present in our environment. So microorganisms such as bacteria, fungi, and algae have shown remarkable ability to transform these pollutants or contaminants present in the environment into less hazardous or non-hazardous substances using their enzymes. Microorganisms can be used to degrade wide range of pollutants such as organic pollutants, heavy metals, and radioactive material. Now let's see what are the advantages and benefits of microremediation. So cost effectiveness. Uh, Microremediation is often cheaper when compared to our traditional cleanup methods. Environmental friendliness. As I mentioned before, microremediation is a completely environmental friendly method because it's a completely natural process where we use microorganisms to degrade these pollutants present in the environment. Here we are not using any chemicals or anything which can have an adverse effect on the environment. And the main advantage of microremediation is that, as you guys know, microorganisms are ubiquitous in nature and they are present everywhere. So the microorganisms will also be present naturally at the site of contamination as well. So what we can do is that we can enhance the conditions at the site of contamination. We can add more nutrients, which will enhance the growth of microorganism, and which will in turn enhance the process of bioremediation. So here there is no need of transferring these contaminated materials or substances into a controlled environment so that they can be treated or degraded. It can be done at the site of contamination itself. Now moving on to the challenges of microremediation. As I mentioned before, they can be performed at the contaminated site itself, microremediation. But here the problem is that as microorganisms are biological organisms, there are so many factors that will influence their activity, such as type of contaminant, site conditions, microbial population, etc. It is also a very time-consuming process. The cleanup can take up, especially large-scale contamination, can take up a lot of time. Monitoring. So, as you all know, microremediation would require regular monitoring. Microremediation of organic pollutants. So, using microorganisms in order to remove organic pollutants, which comes from industries such as oil spills or petroleum industries, is a very effective method. So, the thing with such organic pollutants is that there is a chance they can remain in our environment, or they can persist in our environment for a very long time. So, we can use microorganisms in order to reduce their levels significantly. So, in order to enhance this process, we can use substances such as biosurfactants, which are produced by certain microorganisms such as Serratia marcescens, which will increase the efficiency of removal of petroleum hydrocarbon from environment. Then we can use processes such as bioaugmentation and biostimulation. As I mentioned before, bioaugmentation is addition of specific microorganism, and biostimulation is providing specific nutrients in order to enhance their activity. Biophiles and bioreactors. These techniques can be, uses controlled environments in order to enhance the process of removal of organic pollutants from environments. Microremediation of polycyclic aromatic hydrocarbons. So as I mentioned in the previous slide, such organic pollutants such as PCBs, pesticides, etc., which has accumulated in our environment due to various human activities, are very hard to be degraded by microorganisms. But there has been several reports that these microorganisms, even though it is very hard to degrade such organic pollutants due to their chemical structures, there have been reports where microorganisms have effectively degraded such persistent organic pollutants. Some examples are also given here. For example, a study which was conducted in 2015, where they used bioreactors and they were able to remove 62 to 69% of oil from contaminated soil. And in another study conducted in 2015 itself, where they used bioreactors to remove 48 to 98% of naphthalene from wastewater. And there are also two other studies given here where they used different methods such as land farming and bioventing in order to remove different types of contaminants from different natural environments. Up next, we will be discussing bioremediation of rubber waste and agriculture waste. So the thing with rubber waste is that they are very difficult to break down and recycle, and they cannot be burned as well, because when they are burned, they will release harmful gases, as we all know. So studies were conducted where scientists used different kind of microorganisms in order to try and degrade this uh, rubber waste. So these studies found out that certain species of fungi can be used to effectively remove the toxic part from the rubber waste, and bacteria can be used to further process it. In case of agriculture waste, agriculture can produce organic waste, which can be a problem, but at the same time they can be source of rich nutrients, so that they can be used as manures for agriculture purposes itself, or otherwise they can be converted into other forms of valuable products such as methane gas, um, which is a rich source of energy. So you all must have heard about vermicomposting. So vermicomposting is an effective method where we can process this agriculture waste into valuable products. Microremediation of heavy metals. So heavy metal toxicity can kill most of the microorganisms present at a contaminated site. Uh, but the thing is that microorganisms are often uh, exposed to such extreme conditions such as extreme pH, extreme temperature, etc. Even if most of the organisms can be killed due to such extreme conditions such as heavy metal toxicity, there is also a chance they can survive and develop resistance to such extreme conditions and adapt to such extreme conditions. So, due to this reason, microorganisms can be considered a very good candidate for remediation of such toxic metals due to their strong adaptive capacity. So, microorganisms can interact with such heavy metals in various ways. So, the thing is that such heavy metals cannot be degraded or destroyed completely. but microorganisms can interact with these heavy metals, and they can change their oxidation state, and they can increase their solubility, and they can precipitate these metals in such various using such various methods. We can effectively remove these heavy metals from the contaminated environment using microorganisms. So moving on, now we will be discussing the chemical factors. So chemical factors plays a crucial role uh, in the bioremediation process because they can have an influence on activity and survival of microorganisms. So here, the bioavailability of contaminants and biodegradability of contaminants matters. So in case of bioavailability of contaminants, a efficient contact or proper contact between microorganisms and contaminants or pollutants is required so that these microorganisms can degrade them efficiently. But sometimes the physical state of these contaminants will disallow a proper contact between microorganisms and these contaminants. So, for example, usually microorganisms prefer substances that are in liquid phase. So, solid phase contaminants will cause a problem for these microorganisms. And polar or water soluble contaminants are more bioavailable for these microorganisms to be degraded. So, in case of hydrophobic contaminants, they may require surfactants. So, we might have to add surfactants, which we discussed in the previous slide. The surfactants which are produced by microorganisms into such contaminated sites so that they can degrade such hydrophobic contaminants. Coming to the biodegradability of contaminants, xenophores, which are not usually present in the environment, their structural features might be very different from the molecules which are usually present in the environment of a microorganism. So due to this reason, as their structural features are very different, they will be difficult for the microorganism to metabolize. So usually microorganisms use enzymes such as oxidoreductases, hydrolyses, lyases, transferases, isomerases, and ligases to mediate the microbial bioremediation. Most of these enzymes are very specific in nature. They can only degrade certain specific contaminants or pollutants. Whereas some enzymes such as toluene dioxygenase, they have a broad substrate specificity. They can degrade various types of aromatic hydrocarbons. So we are coming to the final part of this discussion. Here we will be discussing technical and operational factors. In technical factors, we have physical and chemical properties of the environment where the contaminant is present. For example, suppose a contaminant is present in a soil sample we have. So the soil soil overall texture of the soil, moisture content of the soil, pH, temperature, redox potential, everything will have an effect on the process of bioremediation because we are using microorganisms to remove, uh, which are basically biological organisms, which are living organisms, to remove these contaminants from this particular soil sample. So all this will have an effect on survival and activity of these microorganisms. Now, bioavailability of contaminants. This is what we discussed in the previous slide in the chemical factors as well. So as we discussed before, physical state, if the contaminant is not in a bioavailable form, it will be difficult for the microorganism to degrade them. Presence of inhibitors. Suppose there is any kind of inhibitory substances in this soil sample, which will prevent the growth of microorganism, it will be they won't be able to degrade them efficiently. Microbial community. So certain specific microorganisms will be required to remove these contaminants from the soil or contaminants from any particular environment. So if these specific microorganisms is absent, the overall success rate of the bioremediation process will decrease. Similarly, the abundance of these microorganisms, the nutrient availability. Suppose uh, if our environment cannot uh, meet the nutritional requirement of this particular microorganism, it will also affect the overall success rate of our bioremediation process. Oxygen levels of the environment, competition. Suppose there is another species of organisms which is giving a tight competition to our microorganism, which we are using in the bioremediation process. This will also have a negative effect on the success of the bioremediation process. Nature of contaminants. So nature of contaminant, the type of pollutant that is present, and their concentration. If more concentration of pollutant is present, it will take more time, or it will be more difficult for the microorganism to degrade them. If less concentration is present, the microorganisms can easily degrade these pollutants. We have the operational factors influencing bioremediation process. So these are various factors which we can control, or we have to control during the bioremediation process in order to increase its success rate. For example, in technology selection, we have to study the site of contamination and the contaminant that is present there in order to decide whether we have to use in situ uh, method or ex situ method. Suppose if the microorganism which is capable of degrading uh, the contaminant present is already present in the environment, we can go for in situ method of bioremediation. Otherwise, we would have to transfer that particular contaminant from that environment into a controlled environment where our specific microorganism which can degrade this can efficiently grow, survive, and degrade this contaminants. So it will depend from case to case. Similarly, we would have to adjust the pH according to our microorganism that we are using, whether it requires acidic or alkaline condition. We would have to adjust that. We would have to regularly monitor and evaluate the growth and survival of this microorganisms. Nutrient supplements. Uh, Nutrients has to be supplied regularly in order to ensure efficient growth of these microorganisms. We have to control the moisture, oxygen supply. The aeration must be properly done in order for these organisms to grow and survive. So all these factors have to be monitored and evaluated regularly, or they have to be controlled in order to increase the success rate of a bioremediation process. Part, as we strive to heal the wounds inflicted upon our planet through various human activities, bioremediation emerges as a beacon of hope. So, by harnessing to such tiny microorganisms that already exist in our nature, by using them effectively, we can restore the balance of ecosystem and reclaim the contaminated land and pave a way for a more healthier and better future. So let's all embrace the wisdom of nature and embrace bioremediation. These are my references. Thank you.